I want to tell you about another service that I use a lot on the Bacterial and Viral Bioinformatics Resource Center, and that's the metagenomic binning pipeline, where you can take a mixed sample and isolate good quality genomes from it. They call those things bins, so bins equals genomes. And you can use this if you have a contaminated sample that you don't want to have to go back to the lab and re-isolate again, but also you can use it for a mixed sample, like something you get from wastewater or from a clinical sample that you're trying to figure out what's there. The example we're going to use is that same one we've been doing, which was a metagenomic sample from that patient who had had a kidney transplant. So this service uses this metagenomic binning pipeline that was developed by Perello out of Overbeek's group. They're a powerful bioinformatics group at Argonne National Lab, and they first developed this for the Pathogen Resource Integration Center, which is now part of BV-BRC. And this is, it's a kind of complicated pipeline. You can start with reads or you can start with contigs. If you start with reads, it has to be assembled into contigs and it's processed. There are just a number of things that go on, dividing things by 12mers. We talk a lot about k-mers, right? So this one uses two different sets of k-mers, those that are 12 bases long and those that are 50 bases long. And so it gets to the point of binning the contigs and then checking for things, looking for bacterial bins or non-bacterial bins. If it gets to a bacterial bin, it goes into annotation using RAS-TK. You get the final bins. If they're non-bacterial bins, it uses something called check V for the viral annotation. And then you get your final viral bins. As mentioned in the last diagram, you can use sequencing reads or assembled contigs. For assembly, the pipeline uses Metaspades or MegaHit, and for annotation, it uses RAS-TK for bacterial bins and Vigor4 for viral bins. And what the report returns are binning reports for bacteria and viral bins. If the bacterial bins, genomes, are of good quality, they're integrated into the resource, but only the person who submitted the job can see those genomes, so they're private to that person. This is what the service looks like when you submit the job, and this is what you have. It'll return good bins and bad bins. And so what are all these things with good and bad, and what do all these things mean? Well, in the tutorial that's associated with, you can see that they look for things called coarse and fine consistency, completeness and contamination. They'll look to see if the number of hypothetical genes are what you would expect in a genome from the same taxa. They make sure that the contigs, the assembled contigs, make sense for the genome size. You want the genome size to be correct, the N50. It also looks for mean coverage, genes that might be problems, and it looks for the Phe-S gene. The Phe-S is a single protein of reasonable size that all bacterial genomes should have, and if they find it, it gets a yes, and that indicates a good genome. And a yellow score indicates a problematic quality bin. So if I were to go back to this guy has some yellow because it looks like it's not complete. For this particular genome, it's missing some genes, and for this one as well. Next, we're going to submit a job, and then when it returns, we'll look at the results. So join me for the next set in this series. Thanks a lot. Bye.